This is our second podcast in our human genetics series, and I'm presenting this just to provide you with some general background information. We'll start this by considering development. Development is the slow process of progressive change in the descendants of a fertilized egg, which will give rise to a complete human being. And during development, the genome interacts with the extracellular environment to produce changes in the cellular phenotype. And you know from your study of embryology that these changes are orderly and they're time dependent. And in 1926, the great biologist E.B. Wilson said that the characteristics of an organism gradually emerge by the utilization of inherited information that is located on the chromosomes. That's a pretty extraordinary statement to be made so early in the 20th century. And here, if you think of genetics just with a brief historical perspective, we can say that modern genetics began when Mendel's work was rediscovered, probably 1900. Human genetics as a scientific discipline probably took fruit in the 1950s after the Second World War. Clinical genetics or medical genetics, which is a branch of medicine, began in the 1960s, and that kind of encompasses research into genetic disorders and its application in diagnostics and genetic counseling and management. Here are some key terms that I think everyone has heard of in relation to genetics, and I put these in the context of some major things that have happened in the history of science. So we know that, for example, Darwin published his Origin of Species in 1859. Mendel was doing his work in the 1860s, and he came up with the terms dominant and recessive. In the early part of the 20th century, Bateson termed concepts of homozygous and heterozygous, and then you can put this in the context of understanding genotypes and phenotypes. Again, in the early decade of the 20th century, it wasn't until the 1930s and 40s that people realized that DNA carries genetic information. Watson and Crick, of course, did their groundbreaking work and published their paper on the double helix in 1952. And of course, the cell cycle was finally recognized with the Nobel Prize for Hartwell, Hunt, and Nurse in the turn of the 21st century. The Human Genome Project, of course, completed the first draft in 2003. So our first effective definition of a gene, the smallest particle representing one hereditary characteristic, in the early 1900s. That allowed advances to occur in the understanding of genetic processes without requiring any knowledge of what genes actually were or even how they worked. A modern definition of a gene, this is around the time of the 1990s, we think of a gene as a region of genomic DNA sequence corresponding to a unit of inheritance which is associated with regulatory regions, transcribed regions, and other functional sequence regions. If we think about the human genome, we have a haploid chromosome set, and our human genome would consist of our nuclear DNA and our mitochondrial DNA. Our nuclear DNA is relatively complex, whereas our mitochondrial DNA is fairly simple. Our nuclear genome is on 22 autosomes plus the X or Y chromosome, and there are approximately 25,000 genes described in the human genome. That constitutes about 2% of our nuclear DNA. The majority of our nuclear DNA is considered non-coding DNA. Of the 25,000 genes, there are perhaps 22,000 protein coding genes and maybe about 3,000 RNA coding genes. This is not a bad time for you to consider reviewing materials on gene organization, satellite DNA, transposons, structure and function of nucleic acids, and chromatin structure. And you should be able to review these from your coursework in biochemistry, molecular biology, and or cell biology. Now, when we think about genetics, we're generally thinking about it in relation to reproduction and consequences to subsequent generations. But there are, of course, developmental issues that relate to genetics, and we might talk about mitotic inheritance, we clearly know about and talk about stem cells. And if you haven't appreciated this yet, we'll talk about X chromosome inactivation. And here I'm just giving a broad overview because these things will be covered in more detail through our course. But if we think about mitotic inheritance, we can think about differentiated cells like fibroblasts or lymphocytes. And here mitosis produces two identical daughter cells. And if these cells continue to divide, they're going to transmit the same active genes to all succeeding generations. This implies that in differentiated cells, some specialized genes are going to be turned on, while others are going to be turned off. So it's clear that's why a fibroblast is different from a lymphocyte with respect to gene expression. If we think about stem cells, we know that undifferentiated cells can undergo mitosis, but they might produce two different populations of daughter cells. You might have a differentiated cell and another undifferentiated cell. And this cartoon just highlights that the cells might divide and form stem cells. Some of these cells may differentiate to have different potentials. And of course we know about X chromosome inactivation, the so-called lion hypothesis, and this indeed relates to dosage compensation. You may realize that in early embryonic development, one X chromosome becomes randomly inactivated. These two chromosomes are going to have identical DNA sequences, but they're going to have different parental origins. So early in embryonic development, some cells are going to randomly inactivate either maternal or paternal X chromosomes. So human females would, in a sense, be mosaics with respect to which X chromosome is expressed in which cell. And we know that in some cells you find a bar body. The bar body is the representation of the inactive X chromosome. Now, females will produce X-linked gene products in quantities that are relatively similar to those that are produced in males that have only one X chromosome. That's because all of the genes on the inactive X chromosome are turned off, or I should say most of the genes are turned off, and that's going to require special gene products, and we'll talk about that later in our course. If we think about embryonic development, we can think about it being controlled by differential gene expression. That would have genetic factors, and we can think about environmental factors, so-called epigenetic factors. And basically, if you think about embryonic development and differential gene expression, we can say that differential gene expression requires that the right gene be expressed in the right place at the right time. I like to think of this in a simple-minded way. During development and throughout life, genes might come in two varieties. They'll either be genes that are turned off or they'll be genes that are turned on. Developmental genes are turned on early in embryogenesis and are subsequently turned off and generally remain off later in fetal life and postnatal life. On the other hand, tissue-specific genes may be turned off early in embryogenesis, but are turned on at the right time and in the right tissue throughout life, whether it's embryonic life or even through adult life. Besides developmental genes, there are other important genes. You've heard about these in biochemistry and in molecular biology. There are housekeeping genes, mitochondrial genes, tumor suppressor genes, and oncogenes. We've heard of non-coding DNA, which is structural DNA. Some people even think there's junk DNA, DNA that doesn't serve any function. And of course, only about 2% of our DNA is coding DNA. This is a cartoon or a series of cartoons from quite a bit ago, 2003, from Time magazine, but it's just to give you a sense. And you appreciate all of this, how genes get switched on and how they're transcribed. You know about enhancers and promoters, and it's just to let you realize that much of this is in the common everyday language of people, how genes are turned on and how they're not, how they're activated. And it's going to be important for you as physicians to have a good understanding of this, to be able to explain in simple-minded terms to your patients. Something that we're beginning to appreciate is epigenetic inheritance. This is where cellular information other than DNA sequence may be heritable during cell division. So we can talk about modifications to genes other than to changes in DNA sequence. So things like DNA methylation, histone acetylation, RNA interference. These are all things that can affect gene expression and can influence epigenetic inheritance. And again, from the popular press, there's an article in the 2013 December issue of Smithsonian Magazine that talks about toxins that affected your grandparents might actually be manifest in second and even third generations. And then there's an article in The Scientist early in 2015 that talks about social adversity may shape people's immune systems and susceptibility to disease. That's a fascinating article that even talks about how people that were in the concentration camps in World War II could have been under such adverse conditions of stress that it would have negatively impacted their immune systems and could have led to subsequent disease in later generations. We know about DNA methylation, so you've probably heard about this in biochemistry. At 6 weeks of development, there are different sets of globin genes that are expressed as opposed to what goes on at 12 weeks of development. So for example, at 6 weeks the epsilon globin genes are active, the gamma globin genes are inactivated because their promoters are methylated. At 12 weeks the reverse happens. The epsilon globin gene promoters get methylated and the gamma globin genes become demethylated, so the gamma globin genes are active at 12 weeks. We'll talk about genomic imprinting. This is a rare form of gene regulation in which one allele of a set of autosomal genes is silenced. And this emphasizes the importance of both alleles for normal development and function. We'll see some examples of imprinting that relate to some rare genetic disorders as our course progresses. And I think you appreciate from embryology that all cells require both maternal and paternal chromosomes for normal development. Usually we associate genes on the paternal chromosomes with growth, and genes on the maternal chromosomes we usually associate with growth suppression. Now let's put some genetics background into the context of disease. So we know that there can be genetic causes of disease, and these may be able to be understood fairly easily. We know there may be environmental causes of disease, and this may or may not be understood so easily. We also know that there are multifactorial causes of disease. So many diseases have both genetic and environmental factors. We might consider something like a myocardial infarct. So we could think of things like hyperlipidemia, atherosclerosis, plaque rupture, or clot formation, and all of these may lead to a myocardial infarct. So we can think of genetic issues that would relate to lipid metabolism. We can think of genetic factors that might help to maintain the structural integrity of blood vessel walls, or that might lead to plaque formation. We can think of various clotting factors, and all of these coming together may influence a myocardial infarct. There are clearly environmental factors like smoking, diet, exercise, or lack thereof, or potentially infection that might relate to any of these steps along the path to a myocardial infarction. And there are clearly other diseases that can lead to potential problems that might relate to a myocardial infarct, like hypertension or diabetes or obesity. So what I'm trying to suggest, just as some background information, and we'll see this throughout our course, that if you look at etiologies of disease, there may be chromosomal causes of disease, there could be genetic causes of disease that may relate to single gene defects like dominant or recessive or sex-linked conditions. We'll certainly talk about these in our course. There may be atypical or non-Mendelian genetic factors that can relate to disease. We'll clearly talk about mitochondrial genetic defects that can relate to disease. There are environmental factors that can relate to disease. And as we've suggested, many etiologies of disease may have multifactorial causes.